Welcome to Brain Ponderings, your window into neuroscience research with guests that are at the forefront of brain research. My guest today is Sandrine Thuret. She's a professor of neuroscience at King's College London, a very famous college in London. She's head of the Neurogenesis and Mental Health Laboratory, so that's what I'm going to talk with her about today. We'll learn about her work on neurogenesis and its relation to brain function and perhaps risk for different neurological disorders. She's also the co-head, or I guess co-chair of the Department of Basic and Clinical Sciences there. So welcome, Sandrine.
Thank you very much, Matt. I'm so happy to be here and honored for the invitation.
And you're French originally, and
Yes.
you know, I tried to dig out your background a little bit. And
What did you find?
well,
any dirt?
you spent some time in Lyon.
Yes, correct. Yeah.
And I actually had what is she? She'd be a niece. No, what is she? She's heck, I don't even know what her relationship. So my wife's mother, my wife's aunt and her husband lived in Lyon, and they had a child there. And he worked for some solar panel company, you know, solar energy company. So we visited them there and toured Lyon and the old ruins of,
oh, nice.
You know, historical sites and so on.
Did you enjoy some of the good food?
Oh yeah,
yeah.
And you know, one thing I eat whole grains a lot, and I actually like bread. And the bread you get in Europe, anywhere in Europe, actually is a lot better than United States.
Yeah, I think it's all about the crust. That's my theory.
And I'm gonna talk with you about the effects of diet and diet composition on the brain. Okay, so you did your undergraduate and you got a master's degree in Lyon too, is that right?
So yeah, so I did. So I have I have an engineering degree, a bioengineering degree that I did before. That was split between. So I'm from Burgundy, and the start of my undergrad was in Dijon, so in Burgundy, and then it was in Clermont-Ferrand, was this Polytech engineering school. And then I did work for a little bit. I went to Australia. I worked for a year for a company, indeed working, more bioengineering on food, actually, food bioengineering. And then I decided that, okay, maybe there are more interesting things I can do with my science or my life than, making some interesting genetic modify food, which was exciting, to be honest.
Yeah.
And then indeed, this is when I went to Lyon, where I did a master in aging biology, because I was really becoming really interested in the context of aging. And within this master, obviously, we had access to lectures on all type of aging, so including neuroscience. And this is where I got really hooked in neuroscience. And then I went in Germany, in Heidelberg, where I did my PhD in neuroscience, but more developmental biology, because reflecting back, I thought, okay, if I want to understand aging better, I need to step back and understand how the brain develops, to further understand how it ages. So then it was more neurodevelopmental biology PhD, and then when I went to the Salk Institute in the lab of Rusty Gage, where I got hooked on neurogenesis.
And were you there? You overlapped with Henriette von Prague?
Yes, we did a short time, maybe a year and a half. Yes, Henriette von Prague was there. And yeah, we had some great discussion indeed about the work she was then doing on exercise. So yeah.
Yeah, their work on, well, she came to my laboratory.
Yes.
As a tenure track investigator. And so first, when she was at the Salk, she showed that running wheel exercises, it increases the production of new neurons from stem cells in the hippocampus. Then when she came to NIH, she did these nice studies where she worked out like the timing and how the different inputs from various neurons form synapses on the newly generated neurons. So, to try to understand how they're actually integrating into the circuits.
Circuits, yeah, very important work, yeah. Mm-hmm.
Okay, and then, to tell you the truth, so one of the reasons I invited you, because I saw it, and why don't you talk about this now? So I had a graduate student, actually, it was when I was at University of Kentucky in the late 1990s, Jaewon Lee.
Yes,
and
I know the paper, yeah.
And he'd done some study. We were doing these experiments with every other day fasting, food deprivation, and finding it was neuroprotective in different models. And then he found it increased. Actually, okay, I should have you saying this, but
No, you go. It's your paper.
Okay, so these stem cells, they're self-renewing, they divide. And then when they get certain signals, and we can talk about that, they may divide asymmetrically, and one of the daughter cells starts to grow axons and dendrites and becomes a neuron. The other one keeps dividing. And so Henriette and Kemperman too, he was involved in that, showed that exercise seems to increase the proliferation of the stem cells and maybe their integration into the circuits, I guess. And then, but what Jaewon found in my lab is that the intermittent fasting seemed to not have that much effect on the proliferation, maybe a little, but it increased their survival of the newly generated neurons. And then now talk about we didn't do anything more on that till you
yeah,
so talk about your paper in molecular psychiatry.
Yeah, so I came upon your paper by Lee and within your team, and I found that was really exciting, and I thought, okay, I want to dissect a little bit the molecular mechanism to understand, why intermittent fasting is going to induce this increase of neurogenesis. So then we thought, okay, let's reproduce the data and extract RNA from the hippocampus and see what's different within at the neurostem cell level or within the granule cell layer within the hippocampus. So, order some mice and then started to run them just doing indeed intermittent fasting. And then we did see an increased neurogenesis. But then when I was looking at how much food they were eating, what was left, basically, I realized that actually the mice that were undergoing intermittent fasting, they were eating a lot more than what you guys could see in your strain, in your background.
Yeah.
But still, we saw an increased neurogenesis. So then, instead of just to directly go and dissect the molecular mechanism, I step back and I say, oh, this is very interesting. So that means that these mice, in this context from this provider, they are eating a lot less the next day. That means that overall their calorie intake or energy intake was only decreased, let's say, of just 10%. So I'm like, so then my question became, before to dissect the molecular mechanism, was like, okay, is this because they eat, just 10% less, which would be very interesting because, a very small, decrease in calorie would have such this huge molecular and cellular impact, or is it the actual act of the fasting that has this impact? So then, I did a group where we said, okay, let's have a group where we are going to do energy restriction, like control daily, where we weighted, what mice that had ad libitum access, so as much as they wanted, and then match 10% less every day. So putting exactly how much the mice would eat, but 10% less. And then in parallel, we run the intermittent fasting that still ate 10% less, but, every other day, but a lot the next day. And then from this data, we could see that indeed the calorie restriction of 10% had a small effect, but not that significant compared to the fasting. So thinking that, well, actually it is the act of the fasting, or potentially now the refeeding, who knows, that was increasing the overall level of neurogenesis. But we did see a difference in cell proliferation, which was interesting indeed. But overall, we did see, like you saw, an increase of neurogenesis just for the intermittent fasting. So we did see an increase of the cell proliferation for the calorie restriction of just 10% and the intermittent fasting, but we only saw the increase of neurogenesis, like so overall after 4 weeks, when actually the mice were fasted, as opposed to just the 10% decrease. So for us, giving us a hint, okay, the 10% calorie restriction has an effect, on proliferation, but actually the effect on the differentiation and the net number of neurons actually only happen if you have the fasting effect, which we thought was really interesting. And then from that we went on to dissect, looking then finally at the molecular mechanism, where we did see a difference. Like there are some genes that are up or down regulated only in the fasting, so including Closo, which is this longevity gene that we only see in the fasting and not in the calorie restriction. So that's what we saw in our hand. And
you also looked at the performance and learning and memory test.
Yeah, so we did see that the mice had improved retrieval memory. So in the water maze, so we didn't see any enhancement in the learning per se. So they would learn equally, but it was a retrieval task where only with the fasting we saw an improvement. So calorie restriction did not have an improvement, but the fasting were the one that were persevering in the right zone, let's say. And then you can discuss, or is it actually really that they are better at retrieval? People could discuss the value of that. Are you better at retrieval, or you are more persevering and you are like, no, I know the platform is here and I'm staying here? Is it because you learn a behavior that fasting, you just wait and you are persevering in your attempt? So, you could discuss that. Yeah.
Yeah, I think from an evolutionary perspective, it kind of makes sense that if food is scarce, you better be able to remember.
I agree.
Where to go, and this neurogenesis is occurring in a brain region that's critical for navigating through the environment, and importantly, remembering where you went, what you encountered, what food source was there, what hazards.
So, yeah, whereas we think that probably the daily calorie restriction would not have the same effect, because the food is coming every day. It's a little bit less, but it's there, it's coming, so potentially it doesn't have this impact.
Yeah.
So, yeah, I would agree with you.
And I had, you mentioned Closo, so I had Dana Dubal.
Oh, great.
Yeah, just I don't know, I'm losing track of time since I'm retired and getting old, but I think about a month ago or so,
I need to watch
she was
it.
talking about all her work on Closo.
Yes, so
exciting. People are interested in this, anti-aging protein, and she looked at neurogenesis and some other things.
Yes, exactly. And she saw similar things as well, that there is a link. So it's definitely happening. And in this mouse model, we see it is increased indeed after fasting. In human, might not be the same thing. So we have some ongoing study where we did, well, it's not the same protocol as the mice. That's the thing. I think there is probably very thin line where, you try to translate what you found in mice and then have a protocol in human, but it's never going to be the same, because you are not be able to, in this protocol we did like the 5-2. So that means it's not like very strict 24 hours every other day like you can do with the mice. But yeah, we saw, so we are writing up the paper. We saw that indeed there is an increase in certain type of memory related to neurogenesis, but we didn't find an increase of Closo in everyone. So it seems that some people are responder to the intervention and some are not, which is not surprising from some of our other data. But
And in the human studies, there's a lot of genetic variability between individuals. There's sex differences, maybe.
Yeah, so we didn't have enough power to do to separate the sex because it was a familiar, like not a huge study. I think it was around 70 people. But yeah, I totally agree with you regarding the genetic. I mean, we have another paper where we look at nutrient sensing pathway related genes that are controlling neurostem cells, where we found that they are important and change during aging in vitro. And then we did what we would call a back translational study, went to epidemiological data, so big cohort studies, twin studies, where we have diet data and exercise data and cognition data. And we found that the genes we had identified that related to nutrient sensing pathways were very small variations, so SNPs. Actually, they are going to indicate whether, let's say, someone will exercise. And if we have this small variation in these specific genes we have identified, then exercise will slow down their slope of cognitive decline. But then if they don't have these SNPs, then exercise has no beneficial effect. And it's the same with adherence to Mediterranean diet. We found another genes that's related to that. So that means some people will have, will exercise, but it will not have an impact on, slowing down their cognitive decline. Same with, Mediterranean diet, which, overall as big number is beneficial, but at the individual level, you see that some people that had a specific SNP, then responded and some not. And the answer to, the question to that is like, well, what should I do? I mean, then the answer is like, well, just eat healthy and exercise, because maybe you have these SNPs that will make you responding to a good quality of life, to a good
lifestyle. Well, I could ask you this, and then have you had your DNA sequenced to see which SNPs you have?
So yeah, no, we have. So I did 23andMe, so I could extract. It's true, I could extract my data and I could look at it, but I haven't actually. But that's a good question.
Yeah, but
I know
my guess though, even if your genetic makeup is such that you may not respond well in terms of cognition or something to exercise, I wouldn't suggest to people they not exercise.
Exactly. That was going to be my say. Like, I think even if I would discover that I don't have the SNP that would make me a responder, I would still exercise and try to eat healthy, to be honest. So yeah, I agree with you. Yeah.
So most of your studies have been animals. You're doing human work now. Now there is evidence that neurogenesis does occur in humans, adult humans.
Yes.
But then you get the question: you can't really study it in an individual human. You can't quantify. So, well, I'm kind of skipping over, but you're trying to develop some like biomarkers of neurogenesis.
Yes.
Based on, that's pretty interesting, based on circulating factors.
Yeah.
So can you talk about that?
Yeah, with pleasure. So this is really something we are using a lot at the moment in the lab and with collaborators as well, so we were inspired by the work from Tony Viscory, where I'm sure you know well, and this parabiosis experiment back from 2011 that was then at the time led by Saul Villeda, where but I explained where.
And I had him on podcast
There you go.
a year and a half.
Yeah, so
the young blood story.
Yes, the parabiosis with the young and blood, meaning like for your audiences, just to give a little quick background, is that when we fuse the circulatory system of a young and an old, what's happening is that they rejuvenate the neurogenic niche where we are neurogenesis in the old one, and we age the old one. So based on that, I was thinking, I say, okay, so, and then we know that the neurogenesis is highly vascularized. We know that systemic factor like diet, exercise will modulate neurogenesis. And then we have now this evidence from Villeda and Vice-Corey. So the idea was that, okay, we are going to have an in vitro parabiosis experiment. So we engineered a human hippocampal progenitor cell line so that, it's a cell line that's always constant. So this doesn't change. So we can, we engineer it with a CMYC-ER. So basically it's immortalized. So when we add 4-hydroxytamoxifen and growth factors, it will keep referring. You remove it, they differentiate, give rise to neuron, astrocyte, tiny bit of oligodendrocyte. And then, so the first experiment we did is that, okay, this is our constant. So let's compare if we put young serum versus old serum, and then develop a series of readout, obviously linked to the neurogenesis process. So not just how many neurons, but the whole process. So, looking at stem cell mass, specific factors, SOX2, nestin, how many cells die, are they sensitive to oxidative stress? So a lot of a big series of marker. And then what we found in this first paper, in a way, the start was a bit disappointing because we thought, okay, can we replicate what they saw in vivo in the mice? Like, do we get, more or less neuron depending on the age? But we didn't find something that was age dependent. So we did find in general that, okay, if you had the old serum on average from older donor, that was 75 on average, and the young one was, I think, 28 on average, we could see that it did increase more cell death, but we didn't see a difference. And this was all spread out in the old cohort. The young ones was so tight, and the old cohort was all spread out. So meaning that you had some old young, so you had phenotype of old donor that had the same phenotype as young, for example, and then some old that more cells were dying. So that was one of the findings. But then when we looked at, we had data on their cognitive performance. So we didn't see, when you looked at the relation between how much more neuron do you get according to your age, there was no correlation. But then there was a correlation with their cognitive performance within the old group and with the volume of their hippocampus and of their granular cell layer. Oh. So meaning that chronological age in our assay was not related to neurogenesis at all. So, which in a way was not surprising because, again, you think that you have huge variability, right, in people over life. So although the young were so tight, because then you don't have this accumulation of, lifestyle.
Yeah, some individuals are aging well and some not so well.
Exactly. So,
and did you, in these kind of studies, well, first you said the size of the hippocampus, so you did MRI,
yes, yes, we had MRI
imaging.
Data. And so that was proportional to how much their serum were producing new neuron. Yeah. But it was independent of age. That was really, I mean, in a way we were like, oh my God, that doesn't work. But then I said, no, wait a minute. This is exciting because that means, when you move into aging, right, you have this heterogeneity that grows, yeah. And this is, a reflection of lifetime exposure. That could be, diet, exercise. So going back to the drawing board and say, well, can we use this assay then indeed to look at, how lifestyle affect this, and then we had subsequent paper where indeed we look at, how dietary compound, 12 years before have an impact, and how lifestyle like exercise also had an impact. So we are using this assay quite a bit for that in that context.
And you have measurements of like insulin resistance and
so
BMI. Does
yes.
any of those things help separate out, which serum was not more age phenotype versus?
That's a really good question. So within this first study, we didn't have any such data because it was a study it was a series of donor control for an Alzheimer's study. So they were, all cognitively healthy, but they didn't collect this data. But then in the subsequent study where that was really looking at the role of diet on brain plasticity and cognition over time, then we had all this data indeed where we really could dig into that and and control for BMI and all this and all of this measure. So from that we could dissect what people were eating, using food derived metabolite with some expert that actually developed the metabolite marker that are derived from food and can tell us like, yes, this is really a metabolite that derives from this type of food. Having as well, food diary, but also exercise routine. So that was a nice a European consortium across 6 different countries with different cohorts with nice data, where we could see that indeed. So the serum metabolome would mediate together with diet and exercise the risk of cognitive decline and dementia. So over 12 years. So from this essay, and this was a big cohort, like it's 2,000 people.
Okay, so talk about specific dietary, you mentioned the Mediterranean diet. Well, can you just kind of describe what most people have probably heard of it, but just describe what that is, and then maybe you can get into some of the different specific chemicals in the diet.
So our approach was, because the approach of the whole consortium was like, okay, lots of people have tried to do intervention or control trial from single compound, right? So omega-3 fatty acid, does it have an impact, or this, does it have an impact? So our approach was like, well, it's probably not just single factor that will have an effect, but it's probably a composite. So this is where the first part of the study was really indeed to take into account all the diet-related metabolite that we could measure and how this was associated with cognitive decline. And we did that in an untargeted way. So basically it was, full discovery. And what we found in this first paper, which was then independent of neurogenesis, right? It's like, let's just first look at that. And then after we use this serum assay where okay, what is mediating what? So in this first study, we found, so there were 3 different coffee metabolites. Then there were some biomarker of citrus intake, cocoa metabolites, and fish metabolites. So it was not a huge surprise, and really actually a big reflection of the Mediterranean diet that would be, rich in fish oil, for example. But then the interesting part when we, in the paper, we say coffee metabolites, which include some, actual coffee metabolite, but not caffeine, because often people mix, oh, caffeine, this is it, right? No, there's loads of different metabolites when you speak with the expert. And actually what we found is that actually caffeine itself had a negative association with the slope of cognitive decline, but the other coffee metabolite had a positive association. So meaning that caffeine was bad, but coffee is good. So like the take-home message is like, potentially decaffeinated coffee is the best in a way.
But what about, so so green tea also has some chemicals, epicatechins.
Yes.
What about tea versus coffee?
I see. So in our study, we didn't see epicatechin coming up, but possibly because this is a cohort. So the discovery cohort was done in South. So it's called the 3C cohort. So this is a big French studies. Our collaborator were from France, and then this one was done in Bordeaux, which is southwest, where I don't think people drink much tea. They will probably drink coffee. And because this was an aging population recruiting above 60, we don't think either they are drinking energy drink like Red Bull or things like that. So we think that, we are not exactly sure where the caffeine comes from and, how does it differentiate from the caffeine metabolite. But that was an interesting finding. The fish was more like...
I should say, though, that was a little surprising, right? There's some literature suggesting caffeine, well, it definitely can make you more alert in the short term.
Yes, exactly.
But so then what you're addressing is what about like lifelong consumption.
No, exactly. Here, that would be lifelong because we measure, basically, we had the serum and we measure all of that 12 years. So it's a 12 years cohort. So, the slope of cognitive decline,
I see.
Is measured over 12 years. So this is a long-term impact. This is not your, I could show you a coffee here. I have a Coke here. It's not great, right? But I had a long day. So the caffeine, we all know that, it's going to help you to stay sharp, but on the long term, it might not.
But maybe, in these kinds of studies, there can always be like confounds. So I'm thinking, well, what if the people who are drinking a lot of coffee are drinking it because they can't stay awake and their memory isn't functioning as well?
Exactly. So we can never rule out in this kind of because this is an epidemiological study, we could argue right here. So you can never argue all these confounding factors that actually, this is like, an effect from, something is happening. They feel that they are not very sharp. So then they start drinking coffee to stay more focused. So potentially this is like a compensatory effect. And what we see actually is not the effect of caffeine on the brain, but it's, something that's happening in the brain that make people drink more coffee. And on top of that, you have to find out, you have to discuss, people that are fast metabolizer and slow metabolizer. So here we are measuring the metabolite. So it could maybe just due to the fact that the people that show that they are fast or slow metabolizer. Because in this study, we didn't have the genetic for these participants. So
as you probably know, my lab did a lot of different things over the years, but it turns out there's a metabolite of caffeine. Caffeine has a structure actually similar to adenosine, and that's probably why it inhibits the adenosine signaling. But there's a metabolite of caffeine that is similar to uric acid. Uric acid is also a similar structure. And a medicinal chemist in my group, Nigel Gregg, we found that uric acid, which has antioxidant activity, can protect neurons against, we did a stroke model and some other things. And then, but it's not very soluble. That's the problem with uric acid. It's not very water soluble. If it gets too high in your blood, you get these crystals forming and you get something called gout. So Nigel's lab put CH3 groups onto the uric acid molecule. So CH3, the methyl group, makes it more lipid soluble. So it gets in the membrane, gets in the cells. And one of those methylated uric acid derivatives is identical to a caffeine metabolite. And we found that was very neuroprotective.
Interesting. So
we never looked at neurogenesis. If you're interested, Nigel's probably still got some at NIA.
Yeah, that might be very interesting to look at indeed. Yeah, no, for sure. Yeah. So there are things that, this is something that comes out and then you just have to ponder and say, how do you explain that?
Yeah.
So and often there are many, many potential avenues you could look at. But yeah.
So you mentioned the chemicals in coffee. Can you talk a little bit about, there's really good epidemiological evidence that diets with a lot of vegetables and the fiber in the vegetables and fruits, particularly the skins of the fruits and so on, are good for overall health. And you've started to look at effects of some of these on neurogenesis, right?
Yeah. So what we found, so within that big study, so our collaborator within the consortium did all the metabolite, and this is what we found. And then on our hand, we had the same sample, and then we run our neurogenesis in vitro assay where we took the serum and put it on the cells. And then what we could find is that, that actually neurogenesis was mediating the effect of some of these compounds on cognition. And it was as well mediated by exercise. So because we had exercise data within the study, so it was not just the diet, but also exercise came up as, a mediator. And then what came out is that the people that actually were non-exercise or non-regular exerciser, they were a lot more sensitive to, the dietary compound in modulating their neurogenesis and the associated cognition. And then so within this subgroup of non-exerciser, we found that indeed if they had, we found that the increased LDL cholesterol level was associated with a decreased stem cell mass in our assay. So we saw less SOX2. And then we saw that there was a decreased beta cryptoxanthine. That's basically a provitamin A biomarker of fruit and vegetable intake. So this beta cryptoxanthine was associated, so the decrease of that molecule was associated also with a decrease of SOX2. So meaning that if you ate less fruit and vegetable, then your stem cell pool was going down. But that was only true in the non-exerciser. The people who were exercising, that if they didn't eat enough vegetable or if they didn't have this metabolite, this decreased metabolite, didn't have an effect. It only had an effect in the people that were not exercising. So in a way that was quite interesting. So yeah, there are some, food that seems to be important, but it seems depending, what you do in the rest of your lifestyle.
Yeah, so that
and
suggests,
mitigate.
people who don't or can't, they're disabled or something, can't exercise, their brain can still benefit from
Yes,
eating fruits and vegetables
exactly.
and keeping their
Yeah,
brain intellectually engaged.
yeah,
Uh,
absolutely.
and
Yeah.
so there's been a lot of, as you know, there's a lot of food supplement companies that market things that aren't necessarily as fully established to have effects on health, including some on brain health. So do you want to talk a little bit about that? And yeah, there's been, as always, there's a lot of data in animal studies, but of looking at whatever, blueberry extracts or resveratrol from red grapes or sulforaphane from broccoli or curcumin from turmeric.
Yeah.
But there actually haven't been very many well-controlled, randomized controlled, placebo-controlled trials looking at their effects on memory or other aspects of the brain.
No, I think this is what we are missing in the field. Some really well-controlled clinical trials really show, yes, it has an effect. So
And
yes.
sorry to interrupt, but
No?
one of the reasons why, and this is a general problem, is that drug companies aren't going to make any money off of these things that you can always already get at the health food store. And drug companies, for better or maybe for worse in some instances, funds many, maybe most clinical trials of chemical substances. So I think the funding, but NIH does have an alternative, I can't remember what they call the institute, it's an alternative medicine type institute that does fund those things. But I think these are things that should be funded.
I agree.
For a long time, when I was at NIH, I kept kind of pushing the administration to fund a lot of clinical trials of intermittent fasting. And they finally have over the last 10 years. And so there's a huge amount of data coming out on that now. But they haven't really put much money into funding trials of
I agree. It's really tricky to get funding for this kind of studies, and I think that's an issue in general, that prevents the field to show hard evidence indeed. So yeah.
But I should say that there were some early ones. I don't know if you remember, you're not old enough, but there's a ginkgo biloba that used to be a big thing.
Yes.
I'm
Mhm.
a long time ago even, and then ginseng, and there have been some clinical trials that suggest that they do not see any effect in improving.
Yes, correct.
Yeah.
Yeah. So that's why in the design of this consortium, which is not an intervention, right? We look backwards.
Yeah.
Um, uh, we took this approach. This is not a single component. This is going to be many. And on top of that, after we found out, well, it's not even just what you eat. It's your lifestyle in general that's going to have an impact. So then it's really hard to say to people, like, oh, you should take this and you should take that supplement and this one, because I don't think we have enough evidence what would be um, the
Yeah.
perfect mix, because it will depend what else are you doing in your life, right? Are you sedentary? Are you exercising? Are you cognitively active? I mean, there's so many confounder that indeed you need this very controlled intervention trial to have something convincing. So to come back to your question about supplement, I mean, personally, I don't take any. So, except, a vitamin supplement,
Yeah.
but that's it. So, trying to eat healthy, I think it's probably, the best way forward. I mean, I do fasting, by design. Most of the time, most of the day, I mean, I don't have time to have lunch. So I have a little breakfast and then maybe something very late at night, or maybe not at all. Or if I eat something, maybe I come late and I don't have anything. So I think by design, this is what's happening to me.
I don't eat breakfast, and I eat all my food within like 6 hours every day.
Exactly. So I think restricting time eating is one that people can actually do.
Yeah.
When we do some public consultation to speak with people, because I don't know how it is in the US, but now in the UK, the funding body are rightly pushing us to say, well, you have to consult the public and see whatever you want to do, is it achievable, does it make sense, is it what people want. So when you speak to people about exercise, they don't want just to go running, because they say, well, they are not going to hold to that. Like non-exerciser, they are not going to do that. I mean, I love running and I run every day. I run to work. So that's in my lifestyle. But people in general, they are not interested in running. So then if you want to design an exercise intervention, you have to be very inventive and you have to, have a varied program, which is what we have done recently. And people really stick to it and it seems to be working. So consulting the people is good. It's the same with fasting. If you tell them like, you have to, reduce calorie, is really difficult because you think it will never end. Whereas I think the fasting, people are responding better in
Yeah.
this time restricted eating.
Yeah.
People say, well, they see an end to it. Well, if you tell people, oh yeah, you just have to eat less for the rest of your life, they are not going to be happy. Whereas time restricted eating is like, they can see it and say, oh yeah, okay. So yes, I think designing that people can achieve is really important, because there's no point for us to say, to have this perfect design and this is how it will work, and no one is going to do it. So yeah, so we have to find a way that there is some biological validity, but also that's achievable by actually the people who want to take it up to slow down slowly aging
Yeah.
and decrease aging associated disease. Yeah. But because when you look at mouse studies, I mean, I think we wrote a review many years ago where we put all the paper out there that shows that all the mouse model of Alzheimer's disease, if you put them on either intermittent fasting, calorie restriction, restricting eating, it all had a positive impact on slowing down the disease, reducing all the A beta plaque, anything. So basically you could prevent, at least in the mouse, all that happening if, in some intervention. But some of these interventions are so strict that not everyone will be able to achieve it. So I think designing interventions that are achievable, it's an important mission.
Yeah. Let's talk about how what mechanisms might account for the ability of chemicals in plants, phytochemicals, to promote neurogenesis? And my general thinking on this from an evolutionary perspective is that we co-evolved with plants. Plants were here before animals, and actually insects were here before we were. And so from the plant's perspective, they don't want to get chewed up and destroyed, right? And so my impression is that a lot of these interesting chemicals produced by plants, their function is to dissuade or keep insects, herbivores from eating too much, particularly the vital parts. And that one way they do this is they seem to uniformly have a bitter taste. Any of these, sulforaphane, curcumin, resveratrol, epicatechins, well, caffeine, quercetin. So that's one way. Little kids don't tend to like bitter tasting stuff, and maybe that's kind of a protective mechanism to prevent it from eating too much of something that might make them sick. So we've done a lot of work along the ideas that these phytochemicals are causing stress on our cells, and our cells are responding in ways that make them better able to resist stress and function better. So kind of adaptive stress responses. And the best example is with sulforaphane. It's really well established. It will activate a transcription factor called NRF2, which in turn upregulates or induces the expression of genes that encode antioxidant enzymes, that the way that cells normally remove them.
No, I think that's a very good hypothesis and way to think. And then you can also think so if you think about omega-3 fatty acids, which are a different kind, very different, right?
Yeah.
Fish. I think then so the plant, I agree with your theory. For the fish oil, I think there is a directly, impact on, the neuronal membrane. Basically, they just need it. So that I think it's going to be a combination indeed, like stress resistance and actually building the cells. And then, all of this dietary compound will have, different effect. And then when we think about the fasting, it's probably in the same direction. I think there are some cross, when you look at the pathway, what induce fasting versus, for example, epicatechin or resveratrol, it's going to, there will be a cross with sirtuins, as you know really well. So there will be probably a cross between the resistance, resilience, whether it's the fasting or eating plant we're not supposed to eat, indeed to make our cells more resistant.
And as you know, that this idea of optimizing health by challenge recovery cycle, so imposing on the system, in this case the brain, the body, maybe kind of a mild, could be an energetic stress like exercise, or it could be fasting, which is a metabolic challenge or stress, and then a recovery period. And so the idea is that the stress period puts the cells in kind of like conserve resources, stress resistance mode, and activates genes that do those things. And then during the recovery period, resting after exercise, eating after fasting, then the cells go into a growth and plasticity mode. And if you stay in one mode, only one mode, you're too long, you'll get in trouble. Obviously, you can't exercise forever, you can't fast forever.
Exactly.
But if you never fast and never exercise, then the cells are never getting these signals that do good things for them.
Yeah, no, totally agree. Yeah, that's why I was mentioning earlier, is it the fasting or the refeed? But I think you're right. You need this cycle. And I had discussion. I was invited to speak into a consortium working with patients with anorexia, and then I was very, very careful into presenting the data and saying exactly that. And I said, if you fast all the time, you are not going to get an increased neurogenesis. You need a refeeding, to impress onto people that might feel like, oh my God, fasting is great. It's not just the fasting. It's like we can, intermittent eating or time control eating. Maybe we have to reward it in that context. The eating is important, but it's, the time between, you leave between the meal that has an effect. Yeah.
Yeah, the anorexia is it's still it's a little complicated, right? Because it's kind of a psychiatric problem where
Yeah.
the it's kind of an obsessive compulsive type
It's
thing.
nearby.
And so they need a lot of help from mental health professionals, but also people who know something about diet and so
Exactly.
on. And but
Yeah.
I've always thought that kind of the historically conventional way for losing weight is calorie restriction, reduce the amount of calories you eat at each meal. Um, but psychologically, for someone with obesity who's overweight, that's very difficult because then every meal they're kind of obsessing about how much food they're taking in, and they're spending a lot of time ruminating on that, and then
Yeah.
that can get into kind of a vicious cycle.
Yeah, absolutely.
Also, there's hormonal things that change with obesity. You get something called leptin resistance. Everyone's probably heard of insulin resistance, where your cells don't respond to insulin. They can't take up glucose, remove it from the blood. But when you eat a meal, there's a hormone that's released into your blood that goes to your brain and tells you, okay, you've eaten enough, stop eating.
Yeah.
People with obesity, the brain doesn't respond to leptin. And so they perceive that they're hungry when in reality, obviously, they're not starving, right? They're overweight. So once they get into that situation, it takes a lot of time and effort but the good news is you can overcome that with, energy restriction, intermittent fasting, and exercise. And these things take at least a month before you can really change these systems so that they're responding in a more healthy way.
Yeah.
Yeah.
Absolutely. Yeah.
Did you want to talk any more? You mentioned sirtuins. I mentioned NRF2. There's a lot of interesting things on terms of pathways that were actually worked out in other cells, but probably apply to stem cells. mTOR, PGC1 alpha,
Yeah.
AMP kinase. Anything you want to say about those?
Yeah, no, I think one we found really interesting in our data set where I mentioned that we find that small variation in the genes, so SNPs, are actually, modulating or important for how people respond to diet or exercise in connection to cognition. And SIRT1 was one of the gene that came up. So yeah, you can have small variation in SIRT1 indeed, that will have this impact. And then we found some other genes that were more obscure, that no one knows about. So that are interesting for us to dig in. But as a proof of concept that we pulled out SIRT1 in there was really exciting. Yeah.
So you're going to keep the other genes secret for now?
No, they are published. You can dig there. One is called ABTB1, which, like I think there was a link, there is a distant link with the epigenetic clock, potentially. So it's maybe methylated or unmethylated. So, but we don't really know its function. And then there's another one that GRB10 as well, that that's, you know, that we found as well, modulating in the context of exercise, that we found, which is another gene that it is related. So this one makes a lot more sense because it's related to insulin-like growth factors. So yeah, as well, but it's not that it was pulled out in other context. So yeah, so these were some of these 3 genes, but yeah, indeed, the third one was really exciting. We said, okay, that makes sense. Yeah.
Yeah, the growth factors make a lot of sense. So I guess we didn't mention this. There's the neuroprogenitor cells. One of the growth factors that was discovered early on that keeps them in a self-renewing state is called basic FGF or FGF2, fibroblast growth factor 2. And then you mentioned IGF-1. That also has an influence on the stem cells. And then BDNF, which there's a huge literature on, some critical for learning and memory and stress resistance and so on. It promotes the differentiation and survival of the newly generated neurons. And exercise, physical exercise, will boost BDNF a lot. It's the most effective natural way we know of to increase BDNF levels in the brain.
Yeah.
We found that intermittent fasting has a small but significant effect in increasing BDNF. And we found it also increases FGF2.
Oh yeah, very interesting. Yeah, that's right. Yeah. Yeah. Yeah.
Did you allow, oh, it's in the evening there, so you don't have to quit this right now.
No.
Okay.
No. Yeah.
I was going to ask you, what's your interest? Are you working on anything related to neurological disorders, depression, anxiety disorders, Alzheimer's?
Yeah, so we are doing some work. So we have done some work in the context of Alzheimer's disease, where we have used this because, you know, that if you look at the literature, there are data, human data and mouse data showing that neurogenesis is altered very early on, very early stages of neurogenesis of AD, which we think that means that potentially neurogenesis is, you know, part of the disease, or let's say very early on there are changes, whether there are compensatory mechanism, as we were discussing earlier. And then of course, once the disease is really, you know, developed, then you have a crash, decreased neurogenesis, like, you know, like these patients are losing.
But it is interesting that so these newly generated neurons, as you mentioned very early on in our conversation, they become dentate granule neurons.
Yes.
And those use glutamate as their neurotransmitter, and they're a critical, all of our sensory information funnels into these neurons, and that's probably why they're so important in learning and memory. But in Alzheimer's disease, and interestingly in severe epileptic seizures, stroke, these dentate granule neurons are resistant relatively to dying compared to other neurons. So there's something about them that they seem to be
more resilient.
The last thing to go.
Exactly. They are more or less the last thing to go, but nevertheless, within AD, they will go, you know, at one point. But what we see early on is that maybe there is some upregulation of transcription factor that, you know, is going to increase the proliferation of the neurostem cells, and then even their differentiation into neuron. That's what we see with our assay. So we did some longitudinal studies with patients that came to the clinic with mild cognitive impairment, and then some will progress to Alzheimer's disease, and then others will stay stable and never develop Alzheimer's disease. So we took their serum over 6 years and we followed them. And what we can see is that, so we can differentiate between the converter versus the non-converter. And then we can even predict up to 3.5 years before using our assay who is going to convert to Alzheimer versus not. And what we saw is that actually early on, the people who are going to convert to AD, there is an increased cell proliferation and an increased cell death as well. But so you have this happening very early on, 3.5 years before the conversion. So this is, one of the study we have in the context of Alzheimer's disease that we have published as well.
So the ones that do convert, there was the serum from those patients had a stronger effect in stimulating neurogenesis. Is that what you said?
Yes, early
so maybe
on.
it's like it's indicating a stress response, like it's a adaptive, an attempt to replace neurons, even though other neurons in the circuit are already probably gone.
So that's what we think as well.
Yeah.
So it's not useful in the sense that we can't say, oh, we are going to stimulate neurogenesis and stop Alzheimer. It's not going to happen, right? Because there are only so few neurons there. But the assay we are using is interesting as a biomarker, as an early biomarker, because then that gives you time maybe to stratify your patient, do some intervention, not necessarily targeting neurogenesis, but it could be that neurogenesis then is a nice early biomarker that you can use. So using just the serum from the patient.
And to make it clear to the listeners and viewers that there's multiple reasons you're doing this biomarker studies, but in the animals, you can more directly look at neurogenesis by injecting them with a DNA base it's not a natural one that you can detect with an antibody. So you can actually you have to euthanize the animal, make a brain section and stain, and then you can actually count neurons. In humans, you can't look in a human study actually directly at neurogenesis.
Yeah, we cannot, and maybe the listener will wonder. So even at you cannot do imaging as well because this process is too small. You can look at activity, but you cannot say is this a new neuron or not. So that's why this is a kind of a proxy using the serum, putting it on the human cells, human progenitor cells, and see how this is activating the neurogenic process or not. So yeah, that's a proxy.
The first evidence for neurogenesis in the adult brain was they used a radio-labeled thymidine, which is a normal base, but is it possible to do PET imaging with that? Maybe it's just too weak of a signal.
Yeah, it's too weak. So people have tried, but yeah, there's nothing yet. I mean, that would be amazing if this would be developed, but yeah.
And okay, one other thing you mentioned, SIRT1. We did work on another enzyme that has a similar effect on its protein targets, SIRT2 and 3, which is in mitochondria. It's only in mitochondria. And so what these enzymes do is they remove a certain chemical group called acetyl group from certain amino acid on proteins, lysine. Okay, so most proteins have number of lysines in them. And when that enzyme acts to remove that acetyl group, it's called deacetylase, then it can affect the activity of the enzyme. And we found that intermittent fasting increases SIRT3 levels. Have you looked at that in any of your, your cultured, I'd like to see in your cultured cells.
With the fasting serum, we haven't looked at it, but so yeah, I mean, what we would like to do is, if we get the funding. Yeah, so we had a study indeed with the 5-2 diet, so where we had the serum and we did the assay, and what we would like to do is indeed now look at the molecular mechanism. So would like to do single cell RNA-seq of serum from responder to the fasting versus non-responder versus to control, and then I think it would be really interesting indeed if we see that kind of response. So yeah, we need to get the funding for that. because
Oh,
single-cell RNA-seq is really expensive still. So yeah.
yeah. Is there anything else you'd like to mention?
No, I'm just thinking, I think that was a good chat. Yeah, no, I'm just thinking, yeah, so I mean what we've in all the study we have, there's one, where whether we look at different aspect of or different mechanism or different disease where, we have some study on depression, for example, or Alzheimer, but yeah, so there's one common mechanism that seems to always pops up is this neuroimmune interaction and inflammation, and seems to have a big impact. So we are trying now to dissect that, how does it work, and and what's happening to the neurostem cells. Are they changing their role actually in response to that, and could it be an answer? So yeah, so that's what we are looking at, like, from everyone in the lab having their different projects. It seems that there's one, these common things that keep coming back, whether you look at, depression, Alzheimer, or even, healthy aging. Yeah, or intervention that are going to alleviate this, inflammation. So yeah.
Yeah. I just put up my most recent episode. It was with John Cryan.
Yeah, I just saw
Oh, it
before.
did on the gut microbiota and how they affect the brain, but that also affects the immune system and
Yes.
inflammation and
Mm-hmm.
yeah. So, ways to prevent too much inflammation, particularly chronic inflammation,
Yes.
and also, we didn't talk about this, but we focused on neurogenesis because that's kind of one of your main readouts. But neurogenesis only occurs in two brain regions that we know of, the
Yes.
dentate gyrus of the hippocampus and also the ventral subventricular zone, which those stem cells give rise to neurons in your olfactory bulb that you process your smelling information. So most of the circuits in the brain are thought not to be able to be replaced, but nevertheless, there's a lot of good data that exercise, we've done some work with intermittent fasting, look at other circuits, diet, they're undoubtedly having beneficial effects that, more directly on the neurons or the glial cells, which.
Oh, the glial cell, of course, the microglia.
Yeah.
Yeah, we can't forget all that, for sure. Sometimes we have a tendency to be neurocentric, and I have colleagues in my department, right? We do work on glial cells, so we have to be, acknowledging their contribution as well, for sure, of course. Yeah, so we are trying now to design some co-culture model where we are co-culturing. So in our model, we have astrocytes, but we don't have microglia. So now we are designing, co-culture with microglia. So exactly to look at all that. Yeah, absolutely.
Yeah. What about stem cells becoming senescent? I had one of my lab members who actually, she passed away a few years ago unexpectedly, Peisu Zhang, who'd done some studies looking at trying to identify in mouse model Alzheimer's disease and actually some postmortem brain tissue from Alzheimer's patients. Are there cells that exhibit a senescent-like phenotype? So senescence, kind of the classical definition is there's some mitotic cell, some cell that normally divides, and then during aging or maybe even with some disease, they stop dividing and they start producing pro-inflammatory molecules, and they may cause a lot of local inflammation and damage. So there are neural progenitor cells that you focused on, but there are also progenitor cells for the oligodendrocytes that myelinate axons. Oligodendrocyte progenitor cells. There's astrocyte progenitor cells. The microglia thought that the main progenitors are actually in the bone marrow because they're pretty much the same as macrophages. So I should know this because we that was the only study we did on senescence, and we found that the oligodendrocyte progenitor cells seem to have a senescent phenotype in the AD tissue and model. What about dentate gyrus, the progenitor cells and the dentate gyrus cells? Do they exhibit a senescent phenotype?
I don't think people have looked at it, or if they have, they haven't reported it. Yeah. So, which could be possible. Like, people have a tendency not to report negative data when they look for something.
Yeah, either way, it'd be very important, right? If they're not exhibiting senescence, that's maybe good news.
Yeah, definitely. What we know in our hand, it's very hard to induce senescence in these granule cells. We have to try really hard to do that. I mean, we have to engineer them to induce senescence. Like lately, we have used progerin to do that. So we really have to work hard to induce senescence in this cell. But that's a good question. Yeah.
So progerin there's these very rare inherited premature aging or accelerated aging disorders where these kids, by the time they're 10, 12, 15, they're already looking old. They're called progerias. And the progerin is a nuclear, I can't remember. It's a nuclear protein, or not?
Yes. Yeah. So I mean, it really depends where where you look at, but yeah, so it's going to be a membrane lamin as well.
The lamin, yeah.
Yeah.
Yeah. Okay, lots to do. So there's a lot of exciting research going on at the basic level. You're trying to translate some, a lot of people are. Listeners, viewers, encourage your representatives to get more funding for this research.
Yes, I agree. Yeah, definitely. Yeah, absolutely. Yeah.
Okay, great. I hope you have a great summer, Sandrine. It's nice to meet you.
You too.
I enjoyed our
Thank you
chat.
so much for inviting me. It was really nice to discuss with you and a pleasure.
Okay, bye.
Thank you. Bye-bye.